So the only sort of successful treatment for snakebite, as we know, is anti-venom. It's very highly effective at reversing the systemic effects of envenoming, stopping spontaneous bleeding within 15 to 20 minutes, with normal coagulation generally restored within 6 hours, although this can sort of relapse as well in some cases. However, the cons are it's very expensive, the dose efficacy is poor, there's adverse effects, it's ineffective against local effects. There's often problems with availability, and cold chain storage and hospital admission is required, which can be an issue in resource poor settings. So sort of these issues are bringing about research into new developments. So looking at small molecule therapies and biological therapies to sort of combat some of these issues and to just offer an alternative and better treatment. So just kind of stepping aside a little bit, for any sort of new treatments and what's traditional for, say, biologics and small molecules, is to go through this kind of a pathway for regulation and approval by many regulated bodies. So you've got your basic research and discovery, preclinical development, which is typically your, say, pharmacokinetic studies, efficacy studies in animal models, different animal models, to then get sort of a work package going for clinical trials, clinical development, before then going through for the filing and approval of these new therapies. However, as we know, anti-venom is very, very different. So the regulatory process for this is using a mouse model, heavily reliant on this. It's the WHO gold standard, which I'm sure everyone's familiar with and has used a lot. So this is where venom, an above lethal dose, is pre-incubated with a therapeutic for 30 minutes, 37 degrees. Then that combination is injected into a mouse intravenously. And what we do essentially is monitor survival time for 24 hours or 6 or 7 hours, which is another sort of refinement to reduce the time frame. And the main outcome is whether the mouse lives or dies, essentially. Some groups have got inhumane endpoints, so when a mouse shows signs of seizure, paralysis, external hemorrhage, or loss of right and reflex, we have to, especially by UK law, have to take these mice earlier, and that's sort of within our guidelines to get this live or dead sort of outcome. So just to sort of give a schematic, so you get an all death group where you give an above lethal dose to ensure full death. You then give groups of mice, groups of 5 mice, different amounts of anti-venom, and see basically how many die. This then goes into a sort of software that then establishes an ED50 result, which then is used to guide sort of clinical doses and anti-venom labels and things like that. So it is definitely an artificial best case readout. It's got a high harm benefit ratio for the mice. Based on this model in the pre-incubation, we can't look at any pharmacokinetics, and it's also really difficult to know what efficacy thresholds are. So what's the sort of minimum effective dose, when, you know, redosing may be necessary, and what's the most appropriate redose schedule. And this sort of model probably won't be appropriate for regulatory approval for brand new therapeutics, such as novel small molecules or novel, say, monoclonal antibodies. So there's sort of a gap here where, you know, we might be at a bit of a stopgap. And also, obviously, it's not very nice for mice. So what we want to do within this project is whether we can improve the current model. So essentially, it doesn't represent or replicate the real world scenario. Someone would never be bitten directly into the bloodstream by a snake. You've got, you know, venom going into different tissues, muscles, which have a whole different effect on the pharmacokinetics of the venom. That makes treatment responses and getting the kinetics right and dose regime of your therapeutic really difficult. Also, nothing's ever co-administered again, so we can't really look at any sort of pharmacokinetics within this model as it stands, which basically doesn't lend itself to predicting clinical outcomes, again, which is quite important when the sort of new clinical trials coming through for small molecules, for example, borastolid and murine and DMPS. The live dead scoring system, it also doesn't provide a very rich data set. It's very binary. You're not getting, you know, more information on the pathology or whether a treatment's got very sort of low level effects that could be built on. It also doesn't support the syndrome specific clinical trial core outcomes. So looking at INR, fibrinogen, and the mice obviously do endure extreme suffering, which obviously we want to reduce, and there's a push for that globally. So When we look more into, say, what's going on specifically with the venom, so taking Echis, and this is as an example, as this is what we focus the model on, we can look at sort of what the venoms do. So we know there's different proteins in the venom that act on different sort of areas of the coagulation cascade, which is a potential that we can sort of exploit as potentially a biomarker and what we could use in sort of determining any treatment response. So again, when we look in envenomed patients and normal patients, healthy patients, there's some sort of markers. So fibrinogen, prothrombin times, the time it takes to form the clot, and the INR, which is just a normalized sort of level of clotting time. We can see a massive difference between those that are envenomed and those that are normal. So it sort of gives us some confidence or some indication in the starting point as to whether these could be sort of established in a mouse model and something that we could use. So that's essentially what we've gone and done. So there's this paper here. So in the blue line is where patients have been given antivenom. You can see that the fibrinogen level massively increases with antivenom treatment, whereas with the red line, patients haven't been given antivenom, and you can see that the fibrinogen level stays very, very low. So this is essentially something we want to replicate in mice with a much lower dose. We can be able to sort of improve animal welfare and get more data from such models. So essentially what we need is a mouse model that can monitor clinically relevant and systemic parameters over time in the same mouse, so we can get a lot of data from one or a subset of mice, which gives us sort of more information to build that data package for translation. And ideally can be used for determining the efficacy of antivenom and different therapeutics, and is much refined in terms of severity, so the mice aren't suffering. And obviously everything that we want to do, we want it as simple and as cheap as possible, so simple to run. So let's go through our workflow to start with a reduced venom dose. Again, we use Echis romanine venom. So we based our starting dose on a minimum defibrinating dose administered intravenously. We then looked at Venoms, so this is a phenotypic clot properties, looked at point of care devices, so fibrinogen and INR, and compared these against gold standard laboratory coagulopathy diagnostics, just to ensure everything is sort of as it seems. So for those who haven't seen Venoms before, it's elastography and just gives you that phenotypic clot property. So you can see when the clot's forming and how long it takes to clot and how firm the clot is basically. So you can compare normal to any sort of envenomed or blood disorders. So this can look at the intrinsic, extrinsic and fibrinogen only sort of pathways using different activators. And in terms of the point of care devices, we've picked one that looks towards the INR and fibrinogen at these points in the clotting cascade, which is also being used to hopefully inform and validate a data set for snakebite clinical trials. So when we've run these studies, you can see in the diagram you get a big clot essentially when just PBS is injected. So it doesn't take long to clot, forms a really firm clot. When we give the current LD50 dose of venom, so this isn't what would be used in a sort of preclinical antivenom study, this is just the one LD50 dose. Typically they would use 2 to 5 times. You can't quantify anything, it's just a flat line, nothing's clotting, and it's basically, you can't really take much from something you can't quantify. When we administer an 18 times lower dose, you can see that a very, very small clot is forming, so you've got something in the range of something you can quantify as a starting block. So looking just at Rotem, just the clotting time, for example, we've looked at a bit of a range of venom doses. Again, this is intravenous. So it's kind of a nice linear response, which is not all to say. We decided these doses were too low. So as you can see, you're not seeing a massive difference from the normal control. So thinking about sort of statistics, and when we want to look at treatment efficacy, we're probably not going to get enough of a window to have a look at that. These values were slightly too high, so a lot of the sample ranges and time points we looked at were out of range. So again, more like this current LD50 dose where we can't really quantify anything. And also the animal started to show signs of ill health, which is what we wanted to avoid. So we decided to go forward with the 1 microgram dose. So just to sort of go through our study design, so we inject the mice with morphine as an analgesic subcutaneously. We then would take pre-venom sample, so our sort of naive healthy mouse sample. We'd then give the venom, intravenously at this 1 microgram dose, then wait 30 minutes, take a sort of post-venom, our venom baseline sample, and then administer anti-venom after 30 minutes. And essentially what we do is pick multiple time points to take micro samples of blood, so really small volumes, so about 30 microliters, and do these studies for 12 hours to see what's going on and any treatment response, essentially. So looking just at the fibrinogen point of care, you can see straight away a massive decline in fibrinogen. The red line is where it's sort of undetectable on our point of care device. So nothing recovers within the 12 hours, which would be sort of expected and what would be sort of traditional in a clinical scenario. When we give anti-venom at 30 minutes, we do see some recovery, which is a positive sign that we're able to detect something, although it doesn't quite go back to normal. We also see the same with the INR device that we're using. So essentially, excuse the error bars, that goes out of range for a few hours and then returns to normal with venom only. And then when we give anti-venom, we see that it doesn't go out of range at all. It stays fairly normal, what you'd expect sort of a healthy mouse to look like. When we look at the ROTEM, so looking specifically at the extrinsic coagulation pathway, you can see the plotting time over time. So the dash bar is healthy mice, and then you can see just the recovery in the plotting time with anti-venom. You do see some natural recovery with just venom, which is interesting to see, as we're not currently capable of seeing this in the live dead model as it's so severe. And these other graphs just go to show as well how you do see more of a response with anti-venom treatment with ROTEM in addition to the point of care devices. When we look at fibrinogen, which we know is wiped out with envenoming, you can see as well it takes a little bit longer to return to normal, and when in the case of mice that just received venom, you're not really seeing much in the way of that natural recovery. So it's an indicator that this is a good sort of marker to look at any treatment responses. So what we've done to try and test the sensitivity of this model is to look using different doses, so a dose response curve with EpiSub-G. So you can see when we pre-incubate, so it's sort of a mock-up of what would be the traditional WHO model, you see a slight dip and then things return to normal quite quickly. So you're not, again, just reaffirming that you're not getting that much from that sort of model. When we give, sorry for the chaos of this one in different colours, when we give different doses of antivenom, however, we do see dose response. And in these two lines just at the bottom, so the purple and blue, you can see that you're essentially seeing a minimum sort of effective no concentration, which is really sort of important and just reaffirms the sensitivity of this model, again, which is something you can't do in the pre-incubated model. And a similar trend with the INR. So again, you see that things slowly come back in the range in a dose response manner, which is quite nice, giving us that sort of confidence that the model is quite sensitive. What we've also tried to do, so this was just based on intravenous, which, as we know, isn't the most accurate representation of what goes on in a clinical scenario. So we wanted to sort of replicate this in an intradermal and subcutaneous model. So for this sort of study, we weren't really too bothered about the lesions. We were more bothered about whether you could detect systemic envenoming and apply these point of care and ROTEM sort of approaches to have a look at this. And basically what we found was that you can, which is really nice. So you see, as you'd expect, sort of kinetically, when you administer venom intravenously, the fibrinogen is depleted really quickly, whereas you see a bit more of an elongated declining fibrinogen levels when the venom is administered subcutaneously or intradermally, that still is depleted along the course of the study, which we've only done for 6 hours at the minute. And then upon dissection, we get these nice sort of lesions. And so it just shows it's kind of the beginnings of a model, which sort of in addition or instead of an intravenous model, that you could potentially have something where you could look at both the sort of localized sort of lesions and treatment response that way, as well as looking systemically. So it's kind of a sort of 2-in-1 model and a bit more realistic for the translation to the clinical scenario. So we're still developing these models. Our plan is to look at different antivenoms with different levels of published efficacy. We're working with statisticians to define endpoints and put in sort of rigorous statistics to power these experiments properly, and apply these to other venoms, to look at the coagulopathy there, and also look more at these local applications and treatment responses that way. So yeah, thanks to our collaborators, such as LSTM, University of Liverpool, and ICPT. And this is just our article on sort of bringing everything together in a more sort of detailed manner, if anyone was interested. Yeah, thank you.